What is transcription? Another repeat.
Yeah, A.
Yeah, what is translation?
That's going to be RNA to protein.
Yeah.
Yeah.
What is elongation?
That's the assembling of amino acids
Yeah, forming
to
what?
a peptide chain, like a.
Yeah, our polypeptide chain. And what are the name of the bonds that are formed that attach the polypeptide chain together?
Peptide.
Good job. And which amino acid is the start of it? Is designated to the start codon?
Methionine.
Methionine.
Good. What is true about endonuclease? This enzyme begins to degrade DNA molecules at specific internal sites in a nucleic acid or of a single-stranded DNA. This enzyme degrades nucleic acids from one end of the DNA molecule. This enzyme phosphorylates DNA, or this enzyme is responsible for making double helical structure of DNA.
I'm going to go with A.
Yep.
Yeah, that is correct.
And then wouldn't the exonucleases be the ones that start from the ends of the DNA?
Yep.
Okay.
What is the purpose? Why do we have endo and exonucleases? What's their job? What do they deal with?
With mistakes. I don't know.
Yeah, well, that's correct. Yeah, with mistakes, with mutations. So they
saw
deal
you
with
erasing.
they deal with mutations. So anytime, like earlier, right? We talked about what's the name of the type of mutation when one single base pair changes?
Point.
What's that type of mutation called?
A point mutation. Yeah, yeah.
A point mutation. Good. So something like an endo and exonuclease, it depends on the type of mutation and kind of when it discovers that there's a mutation, right? So let's say it's building DNA and it's going, and maybe it's like messed up. It has a single point mutation and it keeps on going. An endonuclease would recognize that, come in, snip out that bad base pair, and help put in the correct one. An exonuclease would be like if we had a shift mutation, like we accidentally got an extra base pair in there. It's like we originally had 2, but then we shoved an extra one in there and it made everything off by 1. An exonuclease might come in and like break and just run from the beginning and just break it all down until it got back to where the mistake is, and it breaks off the mistake, and then the DNA polymerase reruns down to build it properly. So that's the endo and the exonuclease job. Okay, what is true about nucleic acids? They only use ribose phosphates. They are polymers of nucleotides. They are dimers of nucleosides, and they generate high-energy phosphate bonds such as ATP.
Polymers of nucleotides.
Yeah. What does that mean?
I don't.
It's that they're a collection of nucleotides in a sequence.
Poly meaning multiple, polymer, multiple of them. Dimer, it'd be like 2 of them stuck together because dinucleotide is a dimer. Good. Which amino acid has only one codon triplet?
Methionine?
Yeah, methionine. What's that triplet?
AUG.
AUG. I feel like that might be important. Please, for the love, even if you don't remember every single one, like, for the love of everything, remember AUG, please.
Now, some of them have like different combinations that will still produce the same amino acid, right?
Yeah. Let me pull up the, if I can type, geez. You'll see these kind of things. They code for multiple. This is also the beauty of DNA. This is what's like, it's so cool that like the body is prepared, because like mistakes happen, right? So like if we look at something like arginine, right, there are actually constantly mutations in our DNA. The mutations in our DNA is actually how you end up getting different different genes and different expressions, different alleles. So like for example, like when you start talking about like eye colors, how like blue eyes is a mutation, it's a gene mutation. So how that happens is you get these different amino acids, and you can have those point mutations, and a lot of the time, like here, CCU, CCC, CCA, CCG. So no matter what, if this third base pair, that third base can mutate to any of these, and it's still proline. So that's what's super cool about this, is like, even if it's mutates, the proteins will still be functional. Eventually, like if you get enough mutations to change things slightly enough, then that's when you'll start to get those expression of different traits and different things. But the body is super resilient, where our DNA can get pretty jacked up as far as like mutations go, and most of the time it will still work out and be completely fine. So cool. Hey, which amino acid is represented by the start codon?
Signing.
If the horse is not dead, it has been thoroughly beaten. Which end of the tRNA binds a specific amino acid?
5'3" or 5 end.
It's going to be the 3' end.
Okay.
That binds. Let me pull up the picture of a tRNA.
I was listening earlier, but I was also in the middle of a phone call, so I apologize.
No, you're good. You're good. Here we go. So this is the way our tRNA looks.
Okay.
5' end, 3' end. So it's on this 3' end over in here that
Okay.
our amino acid is going to be bound. This is forcing your brains way back. Does anybody remember the mnemonic device to remember the essential amino acids?
Something about living on a branch.
So those are the branch chain amino acids.
Oh, essential amino acids.
Live on a branch is our branch chain amino acids. We have your pal Tyrone trips you in the ring for the ring structures. To be in MCC, you have to have soul. You have your city of ass and glutes, right? All of these guys. If you remember, you have private Tim Hall. Remember that one? Private Tim Hall?
Yeah.
These are our essential amino acids. That's going to come back to bite you guys a lot. If you haven't seen that or looked at it at all, I would make sure the final, I don't think he won't have a ton on there about that specifically, but in future tries, a chunk in biochem 2 later on as well. When you start to talk about some of like the amino acid deficiencies and conditions that revolve around those like dysfunctions and pathology courses, like, and also your clinical nutrition course, like these are going to come back. So make sure you start making like a little folder or something of stuff to review so you can come back to that at some point and look at it again. Which of the following best describes the major components of an initiation complex formed during the process of translation?
Ribosomes.
A lot of big words.
Try again. mRNA and tRNA.
Yeah. So that initiation complex is when we have our tRNA and we have those 2 ribosome subunits come in. What are those 2 subunits referred to as? What are their names?
The 40S
That's
and
the name.
60S.
The 40S and the 60S, and they combine to form what?
80S.
Good. So this tRNA, right? We have our tRNA, we have our ribosomes, and then this is our mRNA. And then our tRNA comes in to here, bringing those amino acids as the mRNA is read, and puts in all of the codons. If you're ever bored and you look up some of the stuff, there's some like simulation videos that they have of this kind of stuff, of like it happening, and it's super, super cool to watch. Where like, actually see if I can find one of them. You have like DNA synthesis in real time. This one, let me see. There's some pretty crazy ones out there where like, might be this one. Let me see. Yeah, like this one. It like shows like the whole RNA polymerase, and then that launches it. So that's like real speed. How fast these things are happening. Isn't that crazy? There's like videos like that you can find them where it's like it shows you the real speed of this stuff happening. Sometimes it's we look at and I say this just because in like the educational side of things, especially next try in physiology, we look at all of this stuff so much as like a step-by-step process. But in reality, this stuff is happening millions upon millions and thousands upon thousands of times within our body in a fraction of the time. So like, especially when you get to learning about the heart, it's so cool. Where you look at like the whole process of like step by step what's happening, and like when certain things get filled, where and how, and what these things like form together, and the sounds that they make because they're doing whatever. And then you realize, oh, like my heart literally beats at least once a second, at all times, 24 hours a day, 365, like, and that is happening nonstop. Like it's so cool. I nerded out a little bit there, but just to, like, take your time to really understand the step by step, but also remember, like, appreciate the reality of what you're learning as well, not just like the nitty gritty details of it. Okay. Question 44. Which of the following can denature a DNA molecule? What does denaturing mean?
Take it apart.
Yeah, rip it apart. So which of the following can denature a DNA molecule?
Alkali?
Yeah, alkali solution. Does anybody remember the pH of blood that it has to, like, the values that it needs to maintain?
7.4.
Yeah,
Isn't it
7.4.
7.35 to 7.45?
Yeah. Any above or below that, and this stuff starts to happen. Which of the following can inhibit translation?
Puromycin.
Yeah. You know what puromycin is?
No, I just know that because of going through these before.
It's another
Yeah.
antibiotic that inhibits protein synthesis. An antibiotic. Have you guys ever taken antibiotics? Done a course of antibiotics?
All the time.
Okay. How does your stomach and gut feel after doing a course of antibiotics?
Gross.
Yeah, because literally antibiotics, while they're killing the infection that's inside of you, they also literally take all, because they don't care. Bacteria is bacteria to an antibiotic. So they jack up your guts. They literally go through and they just kill everything. So it takes a while for your gut to build back up its natural biome. Which of the following chemical bond type is responsible for base pairing between a purine and a pyrimidine nucleotide?
Phosphodiester bond.
Between a purine and a pyrimidine?
Or
So the
is that still hydrogen?
phosphodiester bond, right, is going to connect the base pairs together. This is referring to what's going across to another.
Oh, so it's still hydrogen, isn't it?
Yeah, there are hydrogen bonds. Which of the following enzymes can be used to join two fragments of DNA in the DNA biosynthesis pathway?
Ligase.
DNA ligase. What does DNA helicase do?
Based off the name, I would assume it helps to form the helixes, the turns.
So it's actually, it has to do with, it does have to do with the turn. They are also responsible for unwinding. Earlier we talked about the topoisomerase. This is specifically for preventing supercoiling of the DNA. I don't know if the book mentions that. Have you guys heard about supercoiling before?
No, not that I remember.
Think of DNA kind of like like two strings. If you curl two strings together and like wind them around each other over and over and over again, what happens to, like, at the end of the string or to the middle of the string?
Like balls up.
Yeah, balls up. So as the helicase works and opens and unwinds the DNA, you're gonna start to get that similar effect, that coiling and kind of that bunching of the DNA. That's supercoiling.
Okay.
So the topoisomerase's job is to kind of monitor that, and literally what it does is it will, as it starts to like really like coil, it will literally snip a chunk of the DNA, like make a little snip, so that it like super unwinds, so it goes back to normal, and then it reattaches it. So then the helicase can keep going and doing its job without worrying about creating a supercoil. This stuff is so cool. It's so cool to understand how the body works. Which of the following is a major component of RNA but not DNA? We should know this one. Yeah, uracil. Maybe. Where's the answer? Oh, it's down here. There we go. Which of the following is a nucleotide?
Adenylate?
Adenylate. What are the other nucleotides? We have the adenine, cytosine, guanine, thymine. Remember these guys?
Mm-hmm. So why is this one adenylate and not adenine?
That's what I'm looking at right now.
Because I got it on my quiz too, and he had it as adenylate on the quiz also.
Hmm. Okay. The almighty Google says the following. Adenylate is a nucleotide that contains the adenine nitrogenous base and is coupled with the ribose, with the sugar ribose and the phosphate molecule. Adenosine is a nucleoside with the nitrogenous base adenine coupled with the sugar ribose. And the adenine is a nitrogenous purine base with a double ring structure. So it's like the step by step. So this is one of those questions of more not necessarily which is correct, but which is the air quotes best answer choice. That's why it's adenylate. Because technically all three of these answers are more or less correct. But because the nucleotide, specifically adenylate, is the nucleotide. So the nucleotide has the sugar ribose and the phosphate molecule. The nucleoside just has the sugar ribose. And then the adenine is just that nitrogenous purine or the pyrimidine. So we want to look at that as also nucleotide of DNA. So we have, let's see if we can find the appropriate answers for these other ones as well. So thymine, nucleotide. It's a nucleobase. I'd probably go through the book, just to be 100%, to see where he mentions this kind of stuff and if it gives the other specific names. or you can spend a little bit of time on Google as well, figuring out the exact quote unquote nucleotide versus nucleoside versus the base pair and like the specific names. I don't think he'll hit super hard on on all those questions. If he had it in the quiz and he has it here, it's more likely to show up on the test. But if he doesn't have the other ones on here, it's less likely to show up. So I wouldn't worry too much about it. But it is good to note nucleotide versus side versus just the base. Any other questions about that?
Nope.
Which of the following is common in both DNAs and RNAs? Phosphodiester bonds, adenine nucleotides, uracil and ribose sugars, deoxyribose and ribose sugars. We know immediately there, not that, because RNAs don't have deoxyribose. Adenine nucleotides and ribose sugars.
No.
We're going to have, and it can't be uracil because that's not in DNA. I have this guy, our phosphodiester bonds and adenine nucleotides. Yeah, that's weird that he has, because here he calls it a nucleotide. That's weird. That's probably, that makes me think that this, in all honesty, is probably just a bad question. If I had to, yeah, I would say this just probably isn't a great question.
I kind of just memorized it because I was like, well, I guess that's how he's gonna want it answered.
Yep. That's gonna be the trick. Just memorize that one. And if it pops up, fantastic, you know the answer. If not, it is what it is. Which of the following is generally true about DNA? DNA replication does not use the RNA polymerase so long as the DNA chain. DNA replication does not need involvement of nucleotides. DNA replication does not need ATP, or DNA replication proceeds from 5' to 3' direction at the replication fork.
Five prime to 3'.
That is correct. It's easy to overthink these kind of questions. Most of the time, like what is generally true, I'd look at that one. Look for the more obvious answer choice is what I mean. Which of the following is the correct composition of a nucleoside? We should know this one because we talked about it. Yeah, it's going to be B. It's the ribose or the deoxyribose sugar and the purine or pyrimidine base. It's the nucleoside. What is the correct composition of the nucleotide?
That one would be B.
Yeah, it's going to be just adding in that phosphate group. Which of the following is the start codon? Geez, enough with this question.
He really wants to know that
the horse is glue at this point. Holy crap. Which of the following RNA molecules contains a poly-A tail?
mRNA.
The mRNA. What's the purpose of the poly-A tail?
Beats me.
Oh no.
So the main purpose, there's two main purposes. One, it makes it more stable and prevents degradation. That's the big one. It also allows it to detach and come off of the DNA once it's transcribed, because you get like the A all the way across in a whole, like in a row. It makes it easier there to detach and break off and to separate. So main function is to make it more stable and prevent its degradation. Have Have you guys ever heard the word telomere?
Yes.
Anybody remember what a telomere is or what its job is?
No.
It's been a
So-
while.
Yeah, so at the very end of our DNA strand, right, we had then all of a sudden have a bunch. It literally looks like this. At the end of that DNA strand, it's just a whole bunch of T's, telomerase just shooting off. And that's where we will get that poly A tail from part of it. And the telomere's job is the same thing, is to help DNA kind of structures be more stable. And as we age, and the more times our DNA is replicated, that T chain at the end gets broken off, starts to break down. And that's when like we're more likely to have mutations and stuff like that happen to our DNA. And that's where then that's why as we get older, you start to see more disease, more cancers, that kind of stuff, because that is broken down. And the telomerase is the enzyme that's responsible for maintaining that and keeping that up. That's one of the theories as to why we age, is that we lose that big long thymine chain and the telomerase can't keep up, and that's why our bodies actually age. One of the theories of aging. Which of the following statements, jumping back real quick, did you know that there's like 6 different theories as to why we age and nobody actually knows why it happens?
I mean, that would make sense. I feel like if they actually knew, then someone would have come up with a way to either slow it down or stop it. So,
Yeah, so they kind of have found that different ways, like with aging, like there are ways to improve telomerase function, and it does seem to extend life. A lot of it has to do with lifestyle changes, though. So, good luck making people do that. So,
especially Americans.
yeah,
We're lazy.
everyone in the world is lazy. It's human nature to be lazy. Literally every single cell in our bodies is lazy. It does things that require the least amount of energy to preserve as much energy as possible. That's literally our body's entire job. So it makes sense. It's human nature to be a little bit lazy. We're just energy conscious. That's what it is.
I like that. I'm gonna start saying that instead of being that I'm lazy.
I'm not lazy. I'm energy conscious.
Energy conscious.
Okay. Which of the following statements about tRNA molecules is/are correct? There's at least one tRNA for each of the 20 amino acids. Although composed of a single strand of RNA, each molecule contains several short double helical regions. All the above are correct. The amino acid is always to an A nucleotide at the 3' end of the molecule.
I feel like it would be there's at least one tRNA for each 20. Is that true?
That is for sure true.
That one I feel good about, but I don't know about the others actually.
Yeah, so the answer is all of the above. When we look at the tRNA structure again, this is like the 2D model of it, but there is like mini helices and that happen here and here and here in these hairpin loops. So yeah, this is a better view. See this? Helix there, helix there.
Okay.
See that? And that's why we do these questions, because now you know. You don't know what you know until you don't know what you don't know until you know it. Which of the following statements is true about DNA?
Semi-conservative?
Yeah, DNA replication is semi-conservative.
Okay, man, thank you.
Which of the following sugar is present in DNA but not RNA?
Deoxyribose.
Yeah, our deoxyribose. Which of the following types of bond links successive nucleotides along the leading strand of nucleic acids?
This one's phosphodiester.
Yeah, these are our phosphodiester bonds. Which one of the following is true about the genetic code? It is absolutely identical in all living things. All codons recognized by given tRNA encode different amino acids. Several different codons may encode the same amino acid, or the first position of the tRNA anticodon is always adenosine.
Several different codons may encode the same amino acid.
Exactly. I don't know if he still does this. He used, when I took the class, he was pretty big at knowing like and this was probably back in more of the amino acid one, but like the 3-letter amino acid code.
I don't think
Like-
he's been huge on it.
He hasn't been big on it? Okay. When I was in school, when I was in Tri 1, he was big on these. I think that was more during the amino acid unit, though.
Yeah, I remember him saying during amino acids, like, make sure you know these, but that's, I mean, he hasn't
they
really mentioned it much after that.
And I haven't seen them come up at all in these questions. I'm dying. But yeah, just because I haven't seen it here, so I don't think it'll come back. But excuse me. Which 2 nitrogenous bases are used to synthesize nucleic acids? Choose the correct pair.
Purines and pyrimidines.
Yeah. Kind of a stupid way to remember purines. Purines are a double ring structure, like this. There's 2 rings together. The way that I remember it is it looks like a pair of glasses. And I just picture like a little nerdy kid who's real pure. And that's how I remember that. I don't know.
I love that.
It's or if you think of like an old man, reading with reading glasses. You're just like, oh, little old man. For some reason, I don't know. That's the way I remember purines.
He's
I
so
love
pure.
That's amazing.
So pure. Which type of RNA contains 3 nucleotide anticodon sequence specific for amino acid codons?
Messenger.
It's our transfer our tRNA.
The example of go with my gut and don't change my answer.
Yep. When tRNA has the amino acid bound to it, attached to it, what's the full name of that?
Aminoacyl tRNA.
Yeah, good job. What's the name of the enzyme responsible for binding that amino acid?
The isomerase one. I don't remember the beginning of that.
It's not an isomerase. Remember, because isomerases do what?
Oh, they like rearrange.
They rearrange. So we need to synthesize our amino acid tRNA. So we use a synthetase. So amino acid tRNA synthetase. Last 3 questions. Blank is/are involved in joining DNA molecules together.
DNA ligase.
Yep. Blank is a left-handed double helix. So we talked about B DNA, right? B DNA is our right-handed. So which is left-handed?
Z.
Yeah, our Z DNA. In translation, during the formation of the initiation complex, methionine tRNA anticodon binds to the ribosomal mRNA complex containing the start codon in the P site.
True.
It is true. If I read that and you were like, confused Pikachu phase what?
Yeah.
Let me grab that. There's a nice picture here somewhere. Maybe. Here we go. Boop. We have our 40S subunit. Right, our 40F subunit. We have these different initiation factors. I would review these. I feel like they are a pretty big deal in the book. He talks about them. There's like a whole step-by-step in the book. I'd make sure you go back over this and read that. Make sure you understand that a little bit better. So we have a P site and an A site. So we get our P site right here. AUG starts in that P site. Our tRNA comes, methionine binds nicely right there. And then our 60S subunit comes and binds once that is formed. E site, there's an A site, a P site, and an E site. E site is our exit site. So our new guys are going to come in this way. And this whole thing's going to slide down this way. So it goes from the P site, it slides, another guy comes in and enters the A site, and then slides, transfers to the P site, and another one comes in and it slides and it slides and it slides and it slides and leaves those that amino acid chain growing and growing and growing and growing. I won't get too deep into that because you can go back and watch the videos and review that. Termination of the translation occurs when the releasing factor binds to the stop codon.
Sure.
And that is also true. Once the whole thing is built and we finally get to the very, very end, he used to have stop codon chart. He used to ask questions about that. But which one was a stop? UAA, UAG, UGA, these guys. I don't think he asks those anymore. Typically he'll just say stop. Just like word stop. So yeah, these are our stop codons. And that indicates that that's going to stop there. There's no more tRNAs are going to be coming in. And then we get that the releasing factor comes in and binds the stop codon and lets go of the protein. And then the protein can go, or the polypeptide chain can go and fold itself all up nicely into its secondary, tertiary, and potentially quaternary structure that we learned way back at the very beginning of the try. Okay. We got through all of it. Good job, guys. Crushed it.
Yes, we did.
Any questions, concerns?
I wish there was a way to compile all the questions from like Linus, because I know his final's comprehensive, and just make up a review session to go through just randoms.
I mean, I was planning on putting at least the 2 Quizlets I already have plus this, add this back into it. So
yeah, because you totally threw me, Liz. I'm like, why am I reading these?
My bad.
I'm like, Liz, you're making me panic over here.
My bad.
And someone did link a Quizlet in the group me, I think.
Yeah, that would be me at the beginning of the trimester.
Yeah.
And that one's actually really good.
But I mean, if we want to, I can throw them all in a Quizlet, and we could do it as a Quizlet because you can randomize it on Quizlet.
That would work.